Well, so I know this has been gone over a few times, but would it be all right if you went over epistasis again? I'm trying to.
Epistasis.
Yes,
Oh,
in chapter 9. So there's
yeah.
pleiotropic and polygenic, incomplete in the epistasis.
Okay,
And
yeah, yeah.
I know the textbook answer, but like, yeah.
So I guess to go over the mechanism, we could just see it like there's going to be a drawn out way to, it's going to take me a while, but I think if I could get a visual representation, it might make more sense. So let's just take, let's just say that our eye color, or yeah, let's say our eye color follows simple inheritance. So hypothetically, let's just say you have brown eyes. Oh, I don't need to make it brown though. One. And we'll say that with simple inheritance, brown is the dominant allele. And let's say you mate with someone who has blue eyes. And because it's the recessive trait, it must be, because it is a recessive trait in this hypothetical, you must have little b in order to express that. So this is the way your oops, big B, little b. This is the way things would be represented normally. But let's say simultaneous to this, we also have this other gene behind the scenes. And we'll call this the, I don't know, the do not allow pigment to exist gene. So albinism, essentially. And let's say in order to express albinism, it's a recessive trait. So you have to have little b. So let's say, you're not mating with someone who has albinism, but let's say maybe you're a carrier for it.
Big B, little b.
Yeah, or actually, let's make a different letter.
A, little b.
Yeah. Okay. So this is the way, 25%. This is you, this is the mate. And so, one child has the chance of not even being a carrier. Another child or two children
carry it.
Yeah, will be carriers, and then you have a fourth of a chance of one of your kids having albinism. Now, if this is present, it overrides any. Yeah, it overrides any of the left. Where the hell is that going? Okay, so it would override. Let's say you're supposed to be this, but simultaneous you carry this. Well, you're not even going to have brown hair or brown eyes or blue eyes. You're gonna, well, actually, we'll say this is hair color, not eye color, because there's some funky rules with that. So let's just say this is hair color. Brown hair, yellow hair. I was gonna say blue hair. So regardless of whatever you end up with here, it is overridden by this. Now, let's say, you only have one child and their albinism gene, both alleles are not positive for albinism. Well, then it doesn't really matter. It only makes a difference if you have both alleles saying no pigment should be created. So it's kind of like a secondary gene that just overrides the rules for another gene, in a sense. But I'm not going to go this deep on the test for sure. Yeah, it'll probably be a much simpler question if I were to ask a question about it. All right. Any more questions? I have a few things for you that I want to make sure you guys No, but before I get to that, or maybe I could stimulate some questions. So I wanted to make sure that you guys knew the difference between, or I wanted to make sure that you guys know what homologous chromosomes are. Can any of you guys explain to me what a homologous chromosome is? It could be anything, and then I'll add to it. Any fun facts? Yeah, same. So homologous chromosomes, same one person. I would say same chromosome number. Yeah, so they have two. So for every chromosome you have in all of your somatic cells, you're going to have two homologous chromosomes for that. So two of the same chromosome number. Now, if they have the same chromosome number, that means that these homologous chromosomes are going to be carrying the same genes. So two of the same homologous, or two of the same chromosome number, therefore carry the same genes. Now the alleles might be different. So like, for example, your chromosome 1, let's just say that it's responsible for hair color. Or not the chromosome itself. It contains a gene at a specific loci on that chromosome that is responsible for hair color. Chromosome number 2, chromosome number 3, and so on aren't going to contain that gene. Only your chromosome number 1, and you're going to have two of them, which means that you're going to have two different potential variants. One from your mom, one that you inherited from your mom, and one that you inherited from your dad. So here on the notes, I have homologous chromosomes, two of the same chromosome number, and I'll put in parentheses chromosome, we'll just put number 2, and then we'll even put one inherited from mom and one inherited from dad. I'll just put which carry the same genes at the same loci on each individual chromosome. Okay, so homologous chromosomes, two of the same chromosome number, so like chromosome number 2, one inherited from mom, one inherited from dad, which carry the same genes at the same loci on each individual chromosome. That's how I would define it. And those are going to be the things. So in anaphase 1 of meiosis, your homologous chromosomes are what are actually being separated. That'll probably be a question on there. What, in anaphase 1, what's happening? What's being separated? If you're thinking of mitosis, well, that's going to be your sister chromatids that are separating. But if you're thinking of meiosis, it is the homologous chromosomes that are being separated. Okay. Yeah, so your homologous chromosomes carry the same genes. What are the haploid cells that you get after meiosis? What's another term for them? What do we call them? Yeah, gametes. Nice. Okay. So when you have a chromosome that has already undergone S phase, remember S phase is synthesis. It's the synthesis phase in interphase. And you've duplicated the DNA. So you have twice the amount of DNA still on one chromosome. It is now composed of two sister chromatids, which I hopefully, yeah, I did define that in the follow along video. So, each part of the X, each of these are called sister chromatids, and they're bound together at the centromere. So this is just nomenclature that, just to make sure we're on the same page. Centromere is what connects your sister chromatids together. Also, and I'm going to write this out, on the centromere, I'll put in parentheses, linking sister chromatids together is a location called the kinetochore, which is the binding site for spindle fibers. And remember, spindle fibers, those are going to be the things created by your centrosomes. That's the organelle that creates the spindle fibers, and the spindle fibers are kind of what move the stuff around during mitosis or meiosis. And they're going to be composed of microtubules. So during anaphase 1, remember, you create that tetrad during, actually, you create the tetrad during prophase, right? So the tetrad synapsis happens, which is the coming together of your homologous pairs. And then you have synapsis, or synapsis occurs, then you have a tetrad, and then you move on to, or then you have crossing over, right? And then you move on to metaphase 1, which all of your homologous pairs are lined up down the middle. And then you have anaphase 1. So what happens is you have, a chromosome with 2 sister chromatids, and you have another chromosome with 2 sister chromatids. Each of those chromosomes are a homologous pair that contain 2 sister chromatids each. And so during anaphase 1, you're splitting. What you're doing is you're separating the homologous pair, you're separating the homologous chromosomes that contain sister chromatids. So you're going to have 2 pairs of sister chromatids. Might kind of sound semantic, but that's we got to know the words. Okay, so I've got a question that is a little bit critical thinky. And this is regarding chapter 9. So let's say you have a dude, Bob, whatever, and his parents have widow's peaks. Okay, so, and I'll just type it out here. So Bob's mom, widow's peak. So I'm describing phenotype. Bob's dad, widow's peak. Okay. And then another fun fact. So we need to kind of describe what does it even mean to have a widow's peak? What is a widow's peak? So let's say widow's peaks are the dominant trait. Okay. So we're going to say that we're going to follow classical Mendelian genetics or simple inheritance. So widow's peak is a dominant trait. And what do you have if you don't have widow's peak? You just, you've got a straight hairline, right? So straight hairline is a recessive trait, meaning you have to have 2 of the same allele to express the straight hairline. You only need 1 of each allele, or you only need to be expressing 1 allele of the dominant trait to express the phenotype. Okay, and then surprise, Bob has a straight hairline. So instead of giving you guys the parent's phenotype or genotype, and then having you conclude the potential children, what I'm doing is I'm giving you the phenotype of the child, the phenotype of the parents, and we want to know what the genotype for the parents are. So the way I would do this, so I'm just gonna, the easiest way to do this, especially when it comes to simple inheritance, is just trial and error. So what does it mean to have a widow's peak? And actually, what I can do here, widow's peak is the dominant trait, so that's going to be big W, and we'll just put straight hairline as little w. And we know that because straight hairline is recessive, we can kind of deduce what Bob's genotype is going to be. It must be little w, little w. So in this case, what does it mean to have a widow's peak? So let's just say, one parent looks like that, and the other parent, what else does it mean to have a widow's peak? In order to have a widow's peak, there's only two potentials. You could be homozygous dominant, or you could be heterozygous dominant. But if you're homozygous recessive, meaning little w, you wouldn't even have a widow's peak. And so these are just two potentials. Let's say this is the mom, this is the dad, and as a result, there is a 0% chance of having a child with a straight hairline if one of the parents, or I guess with this combination. So from this point, I'd be like, well, we know that Bob has a straight hairline and his parents have widow's peaks. We need to somehow pass on a homozygous recessive child, or pass on our genes to create a homozygous recessive child. And so after that error, I'd run another trial. Whoa, weird. I was using pencil rather than paintbrush. I'd probably run something like this. Making Bob the 25% chance of getting a straight hairline. So he had a 25% chance of having a straight hairline, and so that would be the only option for that. So kind of critical thinking, you're working backwards a little bit rather than moving forward, rather than working forward. So I'll have probably one question on the test like that. Let's see, what else do we have? When chromosomes are uncondensed, so when they're decondensed during interphase, what would their form be? Do any of you guys know? Chromatin. Yep. Know the word chromatin. That's just the decondensed form. If it's decondensed, more surface area is exposed, and that means that there's more potential for enzymes or other functional proteins to interact with it to help express genes. And that's what you want to do as a cell in interphase. You want to be expressing genes and reacting to your environment. Okay. It's important to know that Mendel tried and succeeded at disproving irreversible blending theory. They'll try to succeed at disproving irreversible blending theory. Okay, that's another fun fact. And remember, irreversible blending theory was this idea that, when you combine two parents and they create an offspring, you will never be able to achieve the same characteristics of your parents or your grandparents for any particular type of trait. And we know the difference between, I'll just type this in for you guys to look up, or know the difference between homozygous and heterozygous. Homo meaning same, hetero meaning different. So if you're homozygous, it means you have two alleles that are the same for one gene. If you're heterozygous, it just means you have two different alleles for the same gene. And the only type of organism that can, or the organism that can only reproduce via binary fission is going to be bacteria. Now there's some short and small cases or outliers where you'll have organisms that are not bacteria that will undergo binary fission, but is not their primary way of reproducing. So the type of organism that can reproduce only through binary fission are going to be bacteria, or archaea too, bacteria or archaea. Prokaryotic cells, anything with a prokaryotic cell, must replicate via binary fission. There's no other option for them. And then know the difference between incomplete dominance, which is going to imply an intermediate phenotype. So you have, red flowers mixing with yellow flowers, you're going to make orange flowers, right? That's going to be incomplete dominance. Co-dominance, blood typing is going to be the prime example, and that's where you have the full expression of both alleles simultaneously. So in the case of flower color, you would have an offspring with red splotches and yellow splotches, rather than an intermediate phenotype, which would be orange. And in the case of blood typing, if you're AA and you create offspring with someone who is BB, so you're hard type A, and you breed with someone who's hard type B, 100% of your children are going to be type AB. But if you're, let's say AO or A small I, and you mate with someone who is B small I. So you're type A, your husband is type B, right? You create a child, you should not be surprised if your child is type O, because you're heterozygous and your husband is heterozygous for type. You're heterozygous for type A, and your husband is heterozygous for type B. So you shouldn't expect for your kid to be type AB, or type A only, or type B only. There is a chance that you will have a child with type O. Know what it entails to work with a sex-linked gene. So color blindness was an example that I talked about. So that's where you will have a gene that is only carried on your sex chromosome. So for example, color blindness is something that's carried on the X chromosome. And you know what, I'll just give an example of that. I'll draw it out. So before we do that, actually, we should talk about, so we could have mom is a carrier for color blindness. And dad is not color blind. And so what letter should we use here? Or actually, we could just use the XY chromosome. So mom is a carrier for color blindness. So what is her, and we could use, we'll use color here. So she's going to be XX, right? So here's one of her alleles that does not contain the allele for color blindness, but she also has another, because she's, she's got two X chromosomes, and one of them happens to carry the allele that says you shall be colorblind. And then we have the dad, which is XY, and he's not colorblind. But remember, and I'll just type this in the notes too, the colorblind gene is carried only on the X chromosome. Okay, I was trying to bold that, but I guess it comes by stock bold. Still stands out. So the colorblind gene is carried only on the X chromosome. And so just to do a monohybrid cross here, you've got XX here. You also have the chance of generating a female that is also a carrier. But then remember, so we got XY here. But remember, your Y chromosome doesn't actually provide any kind of buffer. So it's not like you could be half colorblind. You are simply the lack of the gene. So the only gene that could be expressed is whatever is carried on the X chromosome. So in this case, your Y doesn't help you at all. You're not homozygous or heterozygous for the gene. You simply only have one X chromosome. So therefore, whatever protein contributes to color blindness or mutation in protein that contributes to color blindness is the only one expressed. And so as a result, if you're a male, you have a higher likelihood of getting color blindness if there's a trait that's carried on the X chromosome. So we have 50% chance, 100% chance. So first of all, you have a 50-50 chance of being a male or female. If you are a male, you have a 50% chance of getting it. If you're a female, you have a 0% chance of being colorblind, but you have a 50% chance of being a carrier for color blindness. So you have to think, so here's your females here, and then here's your males here. So 50-50 male or female, and then 50-50 depending on what you are, So if we're looking at it overall, you have a 25% chance of making an offspring that is colorblind, if you're looking at the bigger picture. All right. Okay, I think those are the main points that I wanted to cover. Did I stir up any questions? You guys have any questions? Oh, also know the difference between pleiotropic and polygenic. Here, I'll just type this out. So know the difference between pleiotropic and polygenic. Be able to tell from an example what type of gene you're dealing with. So pleiotropic is going to be, the example of pleiotropic would be one gene creating multiple characteristics. So one gene or allele being responsible for multiple phenotypes. And the example I used in class were Siamese cats, how their cross-eyedness is kind of associated, or it is directly associated with the gene that also contributes to their fur pattern. And then another example was redheads and freckles. Usually people that are redheads will also have freckles because the same gene that controls the pigment also controls the pigmentation of the skin too, including freckles, which is kind of a pigmentation. And then, so that's pleiotropic. Polygenic, so the first part of the word is poly, meaning many, so that's many genes all contributing to one phenotype, all contributing to one characteristic. So this could be height, this could be skin color, and it could even be hair color. We have a few genes that all contribute to even hair color, the type of hair color. I mean, if you're blonde, but there's a spectrum, right? There's a spectrum of brown, there's a spectrum of blonde, there's a spectrum even of having like more or less red hair, and that's because multiple genes contribute to the pigmentation. So we're not peas, we're a little more complicated. You don't have binary height. Okay. And also know, if I'm giving you guys a image of a cell, so we'll just draw a cell, and this cell is so that's going to be your chromosome 1 from your mom. So it'll be chromosome 1 from your dad. All right. Yeah, we'll do with 4. And this is going to be your chromosome number 2 from mom. And this is going to be your chromosome 2 from dad. And as you could probably infer, we're going through some form of anaphase, right? And the form of anaphase that is separating sister chromatids, so it's going to be mitosis. If it were meiosis, I would have probably depicted them in a way where you would see where things crossed over, but I didn't. So this is going to be mitosis. And the general rule of thumb is the way you count chromosomes in a cell is you count the amount of centromeres. So we've got centromere, 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 centromere. So that's 4 centromeres plus 4 centromeres. So if I were to ask you how many chromosomes does this specific cell have, at this moment, it will have 8 chromosomes. Does that make sense? You're counting the centromeres. Now this is going to be the same, or the same kind of rule applies. So like if we were dealing with a cell like this, let's say it was in metaphase. So you've got, and I'll even draw it in a way. So that's your chromosome, right? Chromosome number 1. Oops, I forgot to make this circle brown. I don't want to confuse you. And that's not a circle. Okay. Chromosome number 1 from mom. Chromosome number 1 from dad. And I should have made, actually they are kind of smaller. Chromosome number 2 from mom. And then chromosome number 2 from dad. In this case, you've got 1, 2, 3, 4 centromeres. So in this case, you have 4 chromosomes at this point in time in this specific cell. So I'll just type out 8 chromosomes, 4 chromosomes. That's because there's gray area. I mean, technically when those centromeres are, combined, there's probably a hard line in between them. So you could say, oh, technically, these are separate chromosomes, but we're not as precise at the moment. Maybe as technology grows and we can just look at everything with an electron microscope, Is there a video we can watch on this? There is actually. One of the pre-lecture videos by Amoeba Sisters goes over this concept. I think it's either in their mitosis or meiosis video. So they go over this exactly. But that's all you need to know about this. Just know that when you're looking at any cell at any given time, count their centromeres.
I have a question. So if this was meiosis, the only difference would be that the DNA would be crossed over, so there'd be multiple colors for each chromosome.
Hold on, can you repeat that one more time? I was reading. I'll get to your question, Judah, after this. What was your question again?
If this was meiosis, would the DNA just be crossed over? So that'd be like the only difference?
Yeah. Okay. If we were looking at, like, let's say, for example, this was in meiosis, I would have probably given you an example like this, and I'll just do it really quick. So crossing over would happen between, you know, the homologous chromosome. So on this red chromosome, you'd have a little bit of red, and then you'd have, or orange, and then you'd have like a little bit of red on one of them, right? Because they crossed over and then they separated. You would have, if you were dealing with meiosis with anaphase 2, I don't want to confuse you, but you would have half the amount of chromosomes being separated. But we never deal with, I never draw out all 46 chromosomes. And so I'm only dealing usually with a few. So it's not going to make a difference, but yes, it's going to look the same, only that I would depict it as crossed over. If you're looking at it under the microscope or under an electron microscope, they wouldn't have color. You probably wouldn't even be able to tell, even from an electron microscope, whether it's undergoing mitosis or meiosis. Actually, you might. Actually, you probably would be able to, but there would be different tellers besides just the chromosomes. It'd probably be, you have a little bit to do with like the size of the cell, the shape of the cell, and the density of chromosome, the density of material being moved around too. Because there are less chromosomes, yeah. And then so we have another question, what are the spindles attached to? That's a good question. These are called centrosomes. That's a question on the test. So know what centrosomes do. They're these organelles that produce spindle fibers. Okay. I think that's all I've got. All the main points I wanted to get over to you guys. Do we have any more questions? And good questions, by the way, everyone who's asked any questions. One more thing is know that your cell cycle is a larger umbrella. It's pretty much just the figure, but remember the figure that cell cycle is the overarching umbrella that contains both interphase containing G1, S, and G2 as well. So cell cycle is going to contain interphase as well as mitotic phase and cytokinesis. So all of those phases are contained within cell cycle. And mitosis containing, you know, your prophase, metaphase, anaphase, telophase. Non-disjunction, that's another term. Do we have to get in depth about the phases too? In a sense, yeah, because you have to know, I want you to know what happens in each phase. And I definitely want you to know the difference between mitosis and meiosis. So be able to tell the difference between anaphase 1 and mitosis versus anaphase 1 and mitosis from a picture. but there's only so much you can do without, you know, I don't have you guys write essays, so and most of the stuff's multiple choice. So you're going to be given a lot of material to work with, like images, if I were having you tell me the difference. Also, the extra credit assignment has some stuff. Yeah, the extra credit assignment for this section has some stuff that I'm looking for in the. I want to be able to give you a list of data. So, you know, your homologous chromosomes are separating from each other. I want you to be able to tell me, is this happening in mitosis or is this going to be happening in meiosis? In that case, it would be happening in meiosis because that's the only event where you would have the separation of homologous chromosomes. So do the extra credit assignment, do the Kahoot, and then look over the study guide. All right.